Welcome to LMG Bio channel. Today I'm going to teach you what is Tajima's D and how you can interpret it. So, Tajima's D is basically how genetic diversity deviates from neutrality. And when you look to the formula, basically is the difference between pi and theta. Pi is the count of differences of nucleotides per pair of sequences, and these differences are averaged all over the pairs and all over the sites. And theta, which is Watterson's theta, is the number of segregating sites, so the number of positions in the alignment that have variation, and this number is normalized by a factor that depends on the sample size, but that's not too important in this case. So I'll basically point out two situations. The first one is if you have a bottleneck. If you have a bottleneck, many rare variations are lost by genetic drift. So in this case, Watterson's theta is drastically decreased. But the variants that survive can be intermediary frequency, so pi doesn't decrease as much as theta, so pi decreases, but not as much as theta. So, but in this case, theta is lower than pi. So Tajima's D, if you look to the formula, is higher than 0. On a second case, during a population expansion, the number of individuals increases, so many recent mutations make Watterson theta increase. So new mutations are in low frequency, so pi is low. So theta is higher than pi, so Tajima's D is lower than 0. Now let's think about selection. If you look to purifying selection, purifying selection basically eliminates deleterious mutations, so pi is reduced and theta stays the same. So Tajima's D is lower than 0. In positive selection, positive selection basically creates a selective sweep where variation is wiped out near a benefic mutation, so pi is reduced and theta stays the same, so pi is the same, Tajima's D is lower than 0, just as purifying selection. In the case of balancing selection, So in balancing selection, it keeps multiple alleles in the population, so increases diversity, and there is enrichment of variants in intermediary frequencies. So pi is increased and theta stays the same. So Tajima's D is higher than 0. And that's it. Thank you for watching. See ya.